But we wanted education that really supports the practical application of what our students are learning directly to the students' lives as well as for their professional work. And of high importance is to be able to offer affordable educational programs. So to support your health as well as your thirst for knowledge, Hawthorn offers 2 webinar presentations each month in holistic health and nutrition at no cost because we believe in your commitment to ongoing learning. Well, greetings everyone, and welcome to Hawthorn University's webinar series. You've got Paula Bartholomew here, and I'm just delighted that you've joined me early for just a few announcements because I've invited nutrition educator Mira Desi to be with us. She's going to help us emphasize the importance of healthy lifestyle with whole foods. Mira shares her food news you can use, and with a special focus today on folic acid. Mira is going to join us as a regular guest for this segment in our webinars, because it's not just what we eat, it's also what's in what we eat that matters too. Right, Mira?
Absolutely.
Okay, and then at the top of the hour, we're going to begin part 1 of our presentation on MTHFR and methylation with functional nutritionist Coleen Walsh. So let's get started with the announcements. For those of you here that for the first time, I want you to know that we have muted everyone. We do that for best sound quality. So you can get the best out of the recording and the proceedings. But we always encourage and value your participation, and we hope that you'll post your questions or comments, and all you have to do is type them in anytime during the presentation using your webinar panel, and we'll be posting information to that panel for you throughout the presentation, so pay attention to that as well as you go through, and then we'll be saving time at the end of the webinar for question and answer period. So it's an opportunity for you to post your questions and comments whenever you're ready and not wait all the way until the end to make sure you're in queue and get your questions asked. And just a reminder that our webinars are on the first and third Tuesday of each month, and an important way that you can help us is to complete the survey that we have after the webinar is completed. It's really an opportunity for you to be able to give us feedback on the presentation and to let us know what you'd like to learn more about in upcoming presentations. In our last webinar, it was August 4th, and we had a presentation on the best thyroid diet. It was with Dr. Nicholas Hedberg. And if you missed it, it's recorded, and you can access it anytime from Hawthorn's website, and you can find that under archived webinars. And I want to thank everyone who took the time to fill out the survey, and congratulate Janet Hess because she won the drawing for the copy of Dr. Hedberg's book, The Best Thyroid Diet. So congratulations, Janet. The book is on its way to you. And we're recording this presentation too, and it'll be archived and up on our website in a few days. And just one more announcement. I'd like to share some Hawthorn pride here. The Holistic Nutrition Credentialing Board is a branch of NANP, the National Association of Nutritional Professionals, and they offer a board certifying exam in holistic nutrition. And last year, Hawthorn students took the exam with a 94% pass rate, and that's really good work, everyone. So we have 15 Hawthorn graduates with Masters in Holistic Nutrition degrees that are now board certified in holistic nutrition. It's awesome. And to that, I say, congratulations to everybody that's successfully completed your degree and passed the exam, and congratulations to all of us here at Hawthorn too, because it takes a village to make it all work. But very proud of that. Good for everybody. So that's it for announcements, and I'd like to begin our food news you can use segment. With that, I'd like to say hi to Mira.
Hi, Paula. Thank you so much for having me here.
It's good to have you back. I'm really looking forward to Colleen's presentation, and just really glad you have a few minutes to talk first.
Oh, me too.
And Colleen's here too, so let's say hi. Okay, Colleen's
Hello.
muted.
Sorry, yeah, I was muted for a minute while Mira was talking. Hello, everyone.
Glad you're here. All right, let's start, Mira. If we're going to talk about folic acid, let's start with what is folic acid.
Basically, it is a synthetic form of a B vitamin called folate. Our body uses folate to make DNA and other genetic material. And so folic acid is, as I said, the synthetic form. It's added to food or used as an ingredient in supplements. The challenge is that although the body absorbs folic acid faster than it can absorb folate, it then has to convert the folic acid to folate in order to get it to work.
Mm-hmm. Then why are they adding it to our food?
Well, part of the reason they add it to our food, as I said, it's used to make DNA and other genetic material. Unfortunately, low folate status does cause an increase in congenital disorders, things like spinal bifida, neural tube defects. Mm-hmm. And it's very important for brain development, for overall function, essentially. Back in 1998, the US and Canada started adding it to the food. It's also added in other countries like Australia, Canada, New Zealand, South Africa. And that was to help lower the incidence of neural tube defects. And so it was chosen to add it by the means of fortification. And I'd like to stop for a second and just sort of branch off and say what fortification is. This is when you add something to a food that is not normally present in the food, especially in the quantities that you're adding it. So it's been synthetically added in because it's believed to be something critical to our dietary intake. I will say that since they've added it to the food since 1998, it has resulted in approximately a 30% reduction in neural tube defects in babies. But unfortunately, and Coleen will be addressing this in detail, folic acid is not the best choice for us.
Well, is it just in processed and refined packaged foods?
Well, yes, it is primarily in processed and refined foods. The things that we find it in the most are things like breakfast cereals. Unfortunately, one of the problems with that is some brands tend to overuse it. And so especially with things like bran flakes or other healthy cereals, and that's healthy in air quotes, I just realized you can't see me. And some of these cereals can have 3 or 4 times the amount needed in a serving, and while it doesn't necessarily hurt you, folic acid has a low toxicity level, the problem is taking too much of it can actually impair vitamin B12 absorption in our body. So
that's a problem.
That is a major problem. And, so when you're buying things like rice, pasta, oatmeal, anything made with flour, crackers, cookies, baking mixes, those kinds of things, it has the potential to have folic acid fortification.
So I imagine there's a good number of people on the call that are gluten-free. What should their concerns be?
Actually, the same concern. Just because it's gluten-free doesn't mean they haven't added folic acid to it. And so, as always, you know me, my favorite hashtag is read the label. And so even if you're gluten-free, you're still potentially getting a highly processed, low nutrient food that is fortified with folic acid.
Yeah. We should have a conversation about gluten-free sometime because it's a longer one. For me, gluten-free doesn't mean just replacing grain-based products with gluten-free ones. It's about avoiding those things because they're the problem. Yes. And that the replacement ones have problem and ingredients in it as well. So fruits and vegetables, it's a whole different stream there than when we're dealing with grains. Well, what about foods that aren't processed? Is there folic acid in whole foods?
Yes, there is. And one of the things that we look at is the fact that these foods are not grains and are very wonderful and rich sources of this food and should be included in a beneficial whole food diet. I did look up some specific examples so that I could share them with you. A half a cup of cooked lentils has 180 micrograms of folate, which is the proper form. That's what we want. A half a cup of cooked spinach has 132 micrograms. a half a cup of cooked black beans is 128 micrograms. 1.5 ounces of sunflower seeds turns out to be a nutrition powerhouse with 101 micrograms. Turnip greens, a half a cup cooked is 85, and a half a cup of cooked broccoli provides 84 micrograms.
Okay. Well, you had mentioned folic acid being a problem, but folate is the better absorbed form. Does folate have any of an issue of impairing B12 absorption?
No, it does not, and part of the reason for that is because our body is able to appropriately process it and utilize it. Obviously, there are limits to how much we should get. Adults are generally should top out somewhere around 1,000 micrograms per day, but as you can see from what we talked about in terms of how much is in lentils, it would be very difficult to get to that level.
Yeah.
And I will say, by the way, that number comes from the Linus Pauling Institute, which is one of my favorite resources for micronutrient information.
Yeah, me too. Well, you know, we're about to start our talk with Coleen on MTHFR and methylation, and so with talking about folic acid being a poor form of the vitamin, actually harmful for people with MTHFR, what kind of concerns do we have there?
Well, folate is responsible for the methylation cycle, and Coleen obviously is the expert on that, and we'll be talking about that in her session. When we use folic acid instead of folate, it does not it does not process very well, and so it tends to accumulate in the tissues and create a toxic load, and this then creates a poor methylation cycle. So it's very difficult that we think we're eating something that's providing an important nutrient, but when we take the wrong form, or a form that our body can't process very well, it creates a problem. And as we'll hear from Coleen, you know, the methylation cycle really is the key to being able to appropriately have a number of different vital interfaces in the body functioning the way they're supposed to.
Absolutely. Wow. Really, this has been valuable information. It's a great prelude to Coleen's presentation. So I want to thank you so much for joining us, Mira, and do come back again and keep us informed.
I would love to. Thank you so much.
You bet. All right, everybody, just a heads up here, you can study at Hawthorn and do important work in your communities that really make an impact. For that, it concludes our announcements and our food news segment, and it's time for me to formally introduce Coleen Walsh and begin the presentation.
Hello. Great job, Mira. Thank you so much.
All right. And if anybody else has just tuned in, let me welcome you to Hawthorn University's webinar series, and thanks for joining us. I'm Paula Bartholomew, and I'll be the moderator for the webinar, and together we're going to learn about MTHFR and methylation with Coleen Walsh, because we're all here to broaden our understanding of the MTHFR gene and why it's so important. Because half of the population has a defect in this gene, and even more people have methylation SNPs, or genetic mutations. And this SNP is at a key position in the methylation cycle, and if it's not working properly, it can affect the whole pathway and create symptoms and disease. So it's an important concept and piece here to grasp. And while methylation cycle abnormalities can predispose us to disease, we've got epigenetic factors that can help guide us in our recommendations and our protocols. And Coleen's going to present the basis for understanding MTHFR, methylation, and epigenetics with suggestions for diet and lifestyle practices that will address concerns in this biochemical pathway. And Coleen is a master nutrition therapist. She's a functional nutritionist and owner of Harvesting Nutrition. She specializes in MTHFR and methylation education and client consultations, incorporating genetics with nutrition and lifestyle practices. She's the winner of the 2014 NANP Impact Award for her research and training programs and is dubbed the Methyl Queen by her colleagues. She offers online trainings, teaches, and consults with a broad range of health professionals and the general public on MTHFR and methylation. And she's the author of the forthcoming book, Thriving with MTHFR, a guide for the everyday mutant. And her passion is to educate and support others along with their journey towards health and with empathy and understanding. How lovely. And congratulations on your upcoming book, Coleen.
Thank you so much, Paula. Yeah, it's a work in progress, but I'm hoping that it will give not only hope, but some great information for people with MTHFR and these other methylation SNPs to really live their full life and not let their genetics rule them.
Absolutely. Well, if anybody can do it well, you can. I look forward to my copy.
Oh, thank you. Yes, absolutely.
All right, I want to let you get started because you've got some really important information to share.
Perfect. Well, welcome to MTHFR and Methylation, the big picture, part 1. Just a disclaimer that this is not providing any medical advice, diagnosis, or treatment. And this information is copyright. If you would like to use this as any ways, please contact me or Hawthorn University. Today we'll take a look at the methylation cycle, the very big picture, and we'll delve a little deeper into the MTHFR enzyme. We'll talk about health consequences that can actually come about if you have these SNPs or these genetic mutations, and we'll talk about what we can do about it with holistic nutrition protocols. We'll also talk about one of my big things that I really warn people about is detoxification when you have these SNPs, because our detoxification pathways can be depleted and very slow and lowered. So gentle detoxification, and we'll have time for question and answers at the end. So this is the methylation pathway, or part of the methylation pathway. This is Dr. Ben Lynch from MTHFR.net or seekinghealth.org, and this is his pathway. So you could see that it's pretty complicated, and it's full of several biochemical pathways, and this includes the whole methylation cycle. During this talk, we'll just get acquainted with a bigger understanding of how these pathways work, and we'll start with a bigger picture. This one is Dr. Amy Yasko, and she deals with the autism population and has been working with the methylation cycle for a long period of time. So this is her cycle. We will start with a 30,000-foot view and very low and slow today to get the understanding of the bigger picture. In 2 weeks, I'll be doing part 2, and we can delve a little deeper in some of these pathways. But this is where we'll start today. This is my methylation cycle. And it is a simplified form, obviously, of the other 2 bigger maps. But the methylation cycle has really evolved over the years. It started with this right methionine cycle, and Dr. Amy Yasko kind of put the methionine cycle and the folate cycle together because she saw how important they were together, and they actually use each other and give each other different nutrients. It evolved over the years even more to include the BH4 cycle and the urea cycle, because the researchers really discovered how connected these pathways are and how interdependent they are as well. And so I speak about these rings almost like as Olympic rings. They can be individual circles, but they can also be rings, and together they make something bigger or larger than itself. And we get a better idea of our bodies when we connect these pathways. So you could see that MTHFR is in the center of these pathways. It's a really crucial point. It's not the be-all and end-all of everything, but it's a crucial point and at the center of it all. And we'll discuss a little bit in detail about this MTHFR enzyme a little bit later. So basically, we can think of the methylation cycle that it runs like the mechanics of an old clock or watch. When you take the back of the face of those old clocks, you see all of these gears working and interacting with each other. And one pushes, another pulls. They all have a specific job to do. I think of the methylation cycles running similarly to the mechanics of a clock. And if one section is flawed, then other sections will respond in kind. If one section is faster, then the other sections will get faster, and slower similarly. So if one gear is messed up, if it's having a problem or a reaction, just like a folate cycle, like you see the MTHFR, if that's slowed down or sped up, then it's going to impact the whole pathways. So here we talk about 5 major biochemical pathways within the methylation cycle. And the far right one is the methionine cycle, and that's in orange. And that really processes the amino acid methionine, creates an important nutrient, SAM or SAMe, and then drops down to homocysteine. And homocysteine can be an important nutrient that we do need, but if it's over or an imbalanced, then that homocysteine can create inflammation. It's an inflammatory marker. Also, it pushes cardiovascular disease and even Alzheimer's disease. And that circles back to be remethylated by the MTHFR enzyme and methylfolate back to methionine. And then right to the left of the methionine cycle, in blue we have the folate cycle. And so that processes the folate that we get from our foods and has several different forms of folate and creates the most bioavailable form of folate, which is methyl tetrahydrofolate. Methylfolate for short. And the MTHFR enzyme helps to create that methylfolate. And then in the purple, to the left of the folate cycle, we have the BH4 cycle, or our neurotransmitter cycle. And that neurotransmitter cycle processes our amino acids and creates our neurotransmitters, and even down further into our hormones. And then far to the left in the yellow is the urea cycle that helps us process ammonia, and mainly in our kidneys and in our liver. On the far right, below the methionine cycle, we process the homocysteine, and that's the transsulfuration pathway, or sulfur, where we process our sulfur molecules and one important nutrient like glutathione. We'll discuss a little bit more in detail, but these are the 5 pathways that really bring together to make up the modern methylation cycle of today. So the big idea is each one of these pathways takes place at the cellular level, so inside of the cell. The most basic job is to take the nutrients that we obtain from food and change them into the different forms or different molecules to make them bioavailable and usable to our body in different ways. The functional status, or how well the methylation is working, really determines our resistance or susceptibility to environmental toxins and microbes. And so we'll just really briefly go through like the 5th grade biology. We don't need to remember, but it just gives us a little contents. So on the far left side, we have the cell. In every cell, we have a nucleus. In the nucleus, we have our chromosomes. The chromosomes are organized into 23 pairs, and each chromosome contains a double helix of our DNA. And our DNA is in every cell of our body. It's divided into 2 segments called genes. And those genes, we have 2 copies or 2 nucleotide bases of each one. And when we talk about SNPs, they're basically genetic mutations is another way to say it. That's single nucleotide polymorphism. So it's one change of the nucleotide in the DNA. And of what is traditionally normal, or what we call wild type. So SNPs, for short, are permanent errors in our DNA. It's a point in the genome where there is a single base difference in that nucleotide. It's inherited through our biological parents, so we get one from each of our parents, one from our mother and one from our father for every gene. And most common type of genetic variation, there's about 10 million SNPs in the human genome, and they account for differences among people. So some of that could be even our hair color or personality traits. And then we can get deeper into how, like, we create proteins, how our enzymes are created, or how food is processed or nutrients are processed. And there's a big question out there that the common thought is that most SNPs have no effect on our health or development. And I put a lot of question marks there because I think that's such a blanket statement that people can get into trouble with. We know a lot now, but we're still constantly learning about the genome, and actually in particularly the epigenome, what lies on top and what influences our genes and turns them on and turns them off. So I put a big question mark in there because we're constantly learning how our body is interacting. So again, this is the methylation cycle. Again, you can see the MTHFR enzyme is in that crucial point. So just looking back, each one of these circles has several different steps on the pathway. And there's several different biochemical processes that happen. And so each one of these steps, if you remember in your biochemistry, needs an enzyme to speed up that reaction. It also needs cofactors and coenzymes like B vitamins and, say, minerals like, say, zinc and magnesium are popular ones. And those all come together with a substrate to help make the end product. So every single one of our biochemical steps needs an enzyme to speed up that chemical reaction. MTHFR is an enzyme. So if you remember the ASE or the AZE sometimes, at the end of a long word will really show us that it is an enzyme. So MTHFR is shorthand for methylene tetrahydrofolate reductase. Try to say that 5 times fast, right? And so the ASE is the enzyme. And that helps us really break down different forms of folate in our body. It is a key regulatory enzyme in the folate and homocysteine metabolism. And that homocysteine metabolism is at the bottom of the methionine cycle on the far right of the circles we were looking at. So it is very important in the production of essential nutrients for health. And the reason why is the methyl group. So methylene tetrahydrofolate goes to create the methyl group. And that methyl group is really an important nutrient. And that goes on to help really create crucial nutrients of SAM, S-adenomethionine, glutathione, which is our master antioxidant and our master detoxifier, and neurotransmitters, and even hormones. So it's really one of the first steps to create that methyl group that is so crucial in our body. And so you'll see just a difference here on the bottom right. You see the methylene tetrahydrofolate, which is the substrate. You have the MTHFR enzyme helping to convert and to speed up this chemical reaction to make the 5-methyl tetrahydrofolate or methylfolate, that last step in the folate pathway. And that creates this CH3 right there, which is the little methyl group, which is 1 carbon and 3 hydrogens. And so they call the methylation cycle or the folate cycle the single 1 carbon metabolism. And that 1 carbon is that 1 carbon and 3 hydrogens. They like to call that 1 carbon metabolism. It's very important for the synthesis of nucleic acids for DNA production and repair. It's important for the conversion of amino acids for neurotransmitter production. And we will go into much more detail on that specific role in part 2 in 2 weeks. Also, it's important for the formation and maturation of red blood cells, white blood cells, platelets. It also works along with B12 and other nutrients in that it creates the red blood cells and our blood cells. It's crucial for every cell of our body. And a deficiency will cause certain types of anemia. So folate is one of an important nutrient for several different pathways. And we'll discuss that a little bit when we talk about certain diseases that can be related. So here is another one of Dr. Amy Yasko's methylation maps, and I put all of she put all of these colors. So all of these are different enzymes in these pathways. And again, this is a very simplified form. Although a little bit more confusing than my simple one, but you don't see all of the steps of the pathways. So there's about 10 different steps between this top circle of the folate, which is the second circle to the left, the tetrahydrofolate, to the methyl tetrahydrofolate. So each one of these will have a different SNP, but these are some of the SNPs that have been researched a little bit more than the others and have been shown to be a little bit more pushing some diseases if there are some SNPs in that. So this, just a note to remember that SNPs, just having a SNP, we all have SNPs. And again, that could just relate to our eye color. But they can or cannot be a problem. There's many factors. It's very multifactorial. It depends on your overall health, your nutrient status to see if there's enough cofactors or coenzymes to help these enzymes or these SNPs. And epigenetics is such a big role, like our stress and how we manage that, our diet, our sleep, our other lifestyle factors, our toxic exposure. So just having these SNPs doesn't mean that they're causing a problem. And I think that's such an important thing to remember if we have clients, if we're a practitioner and we have clients, or if we're someone that just got our report and sees all of these SNPs, oh my gosh, what's going on? I'm going to be so sick. We just have to take a deep breath and say, okay, this doesn't mean everything, and diet and lifestyle can really do a lot towards helping these out. So moving on, oops, let me back up one, the methylation cycle. So just a big picture, it occurs billions of times every second in our cells. And each one of these pathways is more prominent in some cells than others. And the folate cycle, though, is in every single cell. And it happens a billion times a second. So it's, as we talked about, it's very important to building neurotransmitters for our DNA, for protein synthesis, as that methyl donor for DNA, for fats, for just so much protection in our body that it could actually manage or contribute to several crucial functions. And that includes detoxification, immune function, energy production, mood balancing, controlling inflammation, or just some of them. But protein synthesis is another reason, major factor in genetic regulation. It also breaks down histamine and processes hormones, especially estrogen. It produces energy, so it's really important to produce other nutrients like CoQ10 and ATP, carnitine, creatinine, and it also produces myelin for our myelin sheath. It helps cell membrane regulation. Coleen is one of the nutrients that is very important in that, as well as building immune cells. So our immunoglobulins, our T cells, our natural killer cells, all of those things. And here's a great little picture of the our drawing of the methyl group that is so important. So we have the one carbon and then the 3 hydrogens. So we just talked about some of these nutrients, the creatinine, the carnitine, the CoQ10, also the phosphatidylcholine or acetylcholine. Glutathione is another major important one. So over here we talked about MTHFR, which is in the blue center, the light blue center. That enzyme helps to produce the methylfolate, and that methylfolate pushes around and it comes around to homocysteine to create methionine. So basically it donates that methyl group, that folate donates that methyl group to homocysteine, and then it turns back into tetrahydrofolate to the top, and it keeps spinning around. That methyl group gets donated to homocysteine, and with B12 as a cofactor and homocysteine along with the methyl group, it turns back into methionine. So it keeps this one spinning around. So the methionine pathway spins around and the folate pathway spins around the opposite side. And so that methionine creates the SAM, which helps to create the CoQ10 and the creatinine and goes over to our Krebs cycle and energy production. And then the phosphatidylcholine actually is down through here in the BHMT. There's other SNPs that come to need that phosphatidylcholine and folate to create the proper forms. And as we talked about a little bit, the homocysteine down and the sulfur nutrients help to create that most important glutathione, but cysteine from homocysteine is right there as well. So all of these nutrients are very important and really crucial to our health, and they come from this methylation pathway. So we'll just talk a little bit what I just said. This is the methylation cycle in motion. It really gives a little bit of an idea, but remember this is very simplified, of what is really happening and how these spin, or these cogs of the wheels, if you remember the back of that clock or the back of that old watch, and these cogs are turning. So it gives you a little bit of an idea of what happens. And I'll say this just briefly again. The methionine takes the amino acid methionine, turns into that most important nutrient, SAM, and SAM is one of the biggest methyl donors, right? So that methyl group, it gives that to DNA, it gives that for energy production. It also comes around and gives it to the neurotransmitters, and we'll talk about that in a couple weeks in depth. And so SAM is an important nutrient. Some people can handle SAM as a supplement, and some people can't. And that turns into SAW, acetyl homocysteine, and that turns into homocysteine. Homocysteine can go 3 different pathways. It can go around, which is basically the long route that happens mostly in the cells, in most of the cells. And that, again, goes with methylfolate. Takes the methyl group for folate, also the B12, which is a couple SNPs here, and that turns into methionine to keep this one spinning around. So you see the red coming around. And then you have the methylfolate that once it donates that methyl group, it turns back into tetrahydrofolate right up that to the top, and then it goes through that biochemical pathway several steps again and keeps spinning. One important thing is B12. If we're low in B12 and we start to give the methyl nutrients to people, that could cause some problems as well. And that could, the deficiency of folate and the deficiency of B12 has really shown to increase homocysteine. And as we know, when that's increased, it's an inflammatory molecule and can push cardiovascular disease and also push Alzheimer's disease. So homocysteine, if this pathway is not working correctly, if it's because of B12, if it's because of cofactors or coenzymes, or if it's because of the MTHFR enzyme is not working properly, if it's slowed down, then there is a shortcut, but that shortcut is not in every cell of our body. So we can't rely on it, and it can be depleted. And that's the BHMT. And that goes through a quicker process. It takes TMG, or trimethylglycine. Coleen is a precursor to that. And it takes that methyl group and makes methionine. The other one way, actually the only one way that homocysteine degrades itself without that methyl group to keep this methionine spinning is through the transsulfuration pathway. And then it drops down, and there's several different nutrients. It gets a lot more complicated, but several different nutrients that it could make, like taurine, one of our big sulfur nutrients, glutathione, which we've talked about, which is just one of our master detoxifiers and master antioxidants, and sulfite. Cysteine is spelled wrong there. I I apologize, there should be an E, and the sulfites down. And there's another SNP there if people are having problems with sulfites. So that just gives us a bigger, clearer picture. Again, a complicated biochemical pathway, but this just gives how they work together synergistically. We'll talk about this more next week, but just briefly since we're here, the methylfolate is needed for this BH4 cycle. It's important to change BH2 into BH4. And BH4 is needed for tyrosine and tryptophan to turn into our neurotransmitters and through the process down here. Also, when we're talking about glutathione and homocysteine, the transsulfuration pathways, SNPs in this pathway can create some higher ammonia. And so that ammonia needs to be detoxified through the urea cycle. And there's certain SNPs that can be slowed down and lowered deficiency with this ammonia detoxification. And then one other thing to even complicate this further is we need BH4 to actually detoxify that ammonia. And we'll get into a little bit of detail. But just another big picture of how complicated these pathways are, but just showing how we really need these to complement each other and to pass nutrients and to actually help detoxify each other in these metabolites that come up. So I'm just waiting for 1 second on my computer. Okay. Methylation can be disturbed by several different mechanisms, as we just talked about the SNPs, having a mutation. If these mutations if we're seeing a lot of patterns, like if you have a different MTHFR enzyme mutations, there's several different ones of those. There's several methylfolate ones that are above that in processing different folates. If there's a pattern, that can definitely cause problems. Also lack of nutrients or even lack of cofactors and lack of coenzymes. So a lot of those cofactors and coenzymes in each of these pathways in the methylation cycle are B vitamins and minerals. So if we have a lack of those nutrients, then those pathways will be deficient whether the SNPs are causing a problem or not. Also, stress can cause so many pathway deficiencies in our nutrients, and so that is related back on. Excessive nutrients can too, like if you have too much of a byproduct, then that can slow down or speed up some of these as well. Environmental toxicity, so there's is very important because there's a lot of different places where different molecules like lead and mercury can stop these pathways in its tracks. So excessive nutrients, end products, environmental toxicity, heavy metals, different chemicals like acetaldehyde, and that can be either from exposure or from some of these biochemical pathways that create these metabolites that our body is not processing well. Also our mental state, like I just spoke about, stress, anxiety, lack of sleep, even being a pessimism. I think Dr. Bruce Lipton, who wrote The Biology of Belief, and Candace Pert, there's so many great authors out there, really do a great job of talking of how we can create big change by the way we think. Methyl medications and methyl depleting nutrients. So certain medications deplete our B vitamins, and especially since we're talking right here, folate and B12 are very important. And they can actually push these pathways further, or they can slow them down. They can have a little bit more toxins in there that our bodies are having trouble pushing through. of those medications can be even antacids or metformin, nitrous oxide, and even high-dose niacin can cause problems with that. And even some of the methyl nutrients, like with some of the different SNPs. Some people still kind of believe the old way of thought of treating MTHFR with high-dose methylfolate and high-dose methylcobalamin or methyl B12. That can be very detrimental to some people as well. So basically, when we look at some of the risks and diseases associated, we really, most studies, scientific studies really focus on what I call the big four that are more prevalent, which is cardiovascular disease, fertility, maintaining pregnancies and birth defects. Third one is cancer. And a fourth one is mood and psychiatric disorders. But there's a close fourth of thyroid and even autoimmune diseases. So it's really important that when we see clients that have a lot of these, then we might want to look into some genetic testing, because they might not be able to handle some of the nutrients as well as other people or medications. So it all breaks down to inability to process nutrients, leading to nutrient deficiencies. And that could even be a problem with our gut and leaky gut or intestinal permeability, or how our digestive system is breaking down foods. But that's one of the first steps, and then it goes into our cells and how our cells and how these biochemical pathways can break down the food. So methylation really helps to turn one nutrient into another and make them more bioavailable when how our bodies need them. So it's very bio-individual. So I like to say that genetics are not the only thing. Genetics load the gun, and our environment pulls the trigger. So we really have a lot to do, especially as nutritionists, and Hawthorn's great program, how we can do that is to help our clients really with these epigenetic factors, and help with our cleaning up our environment. So these epigenetics is the science of how our environment really triggers, influences, and modifies and regulates our genetic activity. Epigenetic triggers not only alter the way our gene code is expressed or not expressed, but there's many epigenetic triggers, and these inheritable changes in genome function that actually occur without a change in our DNA sequence. So I love this picture on the right, where we really do have a lot of power with our DNA and how we can influence our genes. And I'm a perfect story, and I have several clients that are great stories, that I was very, very sick several years ago. And once I started changing my diet, reducing my stress, really changing a lot of the lifestyle modifications, and taking out what Mira was talking about, like some of those synthetic folic acids that are in our food, and then supplying my body with supplementation and nutrients that I did need, I turned my health around. It's going to be a lifelong process, but I feel so much better. So doing these holistic nutrition protocols, we have so much to offer our clients, and it's a multifaceted approach. And it's definitely needed for a lot of our very sick or ill clients. But even our clients that may just be starting illness or disease, and we can really start to help them protect their genetics. And so diet and lifestyle should always be first. And these things can turn a client's health around. We really do need to start this first. Improving gut health, as we know, if we start with the food and they have good quality whole foods coming into their diet, but they're not being able to absorb them or digest the food properly, then that's a big problem as well. if we clean up that dietary, we clean up the lifestyle, we clean up the gut, then it goes into the cell. And restoring our cell membrane, our mitochondrial health, our adrenal health, right? Correcting any blood sugar imbalances, nutritional deficiencies, right? Even if the gut health is cleaned up, maybe they're not processing these nutrients, and that's when we can start to look at these SNPs or genetics. And another important part, improving our detoxification ability pathways, and making sure that we do this very gently and methodically and slowly. Also, reduction of inflammation and oxidative stress, as that can create a lot of stress into our bodies. And some of these methylation pathways can, if not working properly, can increase inflammation and oxidative stress. So one of the big things I really wanted to say today is a warning for detoxification. There are several reasons why glutathione depletion is very relevant in these methylation cycle defects or SNPs. So not having that master antioxidant and that master detoxifier will lower our detoxification ability. But even diet can cause some detox reactions in some people. So even taking out some of those nutrients, we want to go low and slow and make sure we're supporting them along the way. There's many variables, so we want to look at all factors. And I always make sure that I tell my practitioners that you really want to start one thing at a time. I would not go into high-dose supplementation, especially with people who are sick. And if you have done a very good intake forms and you've asked them a lot of questions, that's my first thing. See where their exposure is. See where their detoxification ability is. How are they even doing with simple things like smelling perfumes or going into a store and smelling off-gassed material? If they're having problems with that, we already know they have a pretty big toxic load. So we need to go pretty gentle, because we live in a very toxic world, and we want to really find the full history and get a good questionnaire of what is happening with our clients before we even start this. So again, a very gentle, slow detox is very important. There's many nutrients that in phase 1, phase 2 liver detoxification are either created or needed with methylation. So there's a lot of back and forth with those nutrients, not only glutathione and our B vitamins and our amino acids. So here's just one study. The ability to eliminate undesired compounds, however, depends completely on the physiological functioning and biochemical status of the individual. Anything that impairs full functioning of detoxification biochemistry, such as nutritional deficiencies, will preclude proper elimination of toxic substances. It is imperative that health providers understand the fundamentals of detoxification physiology and biochemistry to secure functioning of the organs of elimination. And so it goes beyond the phase 1, phase 2 detoxification, because we need all of those nutrients for those. And I really caution people, make sure that you're individualizing. No detox in a box, because if we're dealing with sicker individuals, then that could be really damaging. So everyone is unique. Therefore, each detoxification should be unique. Some things that we can start first for detoxification while working on diet and lifestyle is reducing the toxic exposure, and that's always the first thing. Determine the toxic load and then begin to slowly eliminate those accumulated detoxins and toxins. So there's so many things that we can do with this, but basically I have this picture of the baby standing, right? before we even take baby steps, I want you just to remember that we need to crawl first. And some clients need those baby steps in even more of a crawling stage. So again, cleaning up the food, the environment, water, air, relationships, attitude, not forgetting those important aspects of our mental and our emotional and our relationship. Tons of alternative therapies can help, but I'm sure each one of you has their bag of tricks to do that with. Supplementation is another warning that I'm going to give you today. We really need to take caution and take aware, and I briefly touched upon that. Like some people cannot handle methylfolate because they are too high in methyl nutrients, and especially COMT, which is in the BH4 cycle. And it processes dopamine, and dopamine has methyl nutrients in it, and they have a reduced capacity for that. So they cannot take too many methyl donors. Other people that cannot do that, high inflammation or oxidative stress. So we really have to go low and slow. So it needs to be individual. So you want to evaluate the individual, their genetics, their history, their symptoms, and the whole person. There's not one protocol that fits all. It's very multifactorial, and supplementation can really push detoxification. So I just have a take-home note here, and I love this by Dr. Lisa Schuler. It was their book Five to Thrive, but now it's called Definitive Guide to Thriving After Cancer. They have a couple different books, but they say we cannot change inherited genetic mutations. We can, however, utilize our lifestyle to minimize exposure to further mutations and, importantly, to alter how our genes behave. Thus, to understand how to alter how our epigenome is to understand how to best prevent cancer. And I would add on not only cancer, because they're talking about cancer, but all other diseases as well. And so we really can do a lot to help our clients. Here is a couple of my favorite resources. Dr. Ben Lynch, he has a couple different sources there. We have Dr. Amy Yasko, has a lot of her methylation cycle, her books for free there, and Dr. Rich Van Cronenberg is a biochemist, does a lot with chronic fatigue. Also, Gene Cards is a great way to do individuals, and Snippedia, you can really do individual SNPs to do some more research on each one of the SNPs if you're having problems. So I can be reached at different ways. I think we're going to go into questions and answers right now, but I do have a practitioner training that goes a little deeper. It's still a 101 and 201 into these biochemical pathways. Both Dr. Yasko and Dr. Ben Lynch have even deeper trainings along with that. So Paula, is there some questions?
Oh, you bet, Coleen. Thank you very much for the presentation. You raise a lot of concerns and paint a really good picture of this. Thank you.
Thank you. Yeah, it's hard to do in a 45-minute presentation, so I'm glad we'll get together in a couple of weeks.
No, it'll be good. Yeah, we'll talk about it even more. So, and a couple of things about testing. Our question is, when or how would you know you need genetic testing, or that somebody would? Are there some signs to look for?
Yeah, I would say, like, if they have the big four, and I know I glanced through that because I was trying to kind of give a big picture of everything, but like cardiovascular, like the four top ones, that's the diseases that are really associated: cardiovascular disease, fertility, miscarriages, pregnancy, all of that, of course, because of the folate and B12 needs. Cancers are very prominent, and so are mood and psychological disorders. But thyroid problems are very close, and so are autoimmune. So if you have a history of all of those things in your family, and I would suggest you trying to talk to your doctor. about getting the test. If they will do it, they'll probably do just the MTHFR alone. And as you know here, it's not only about that. 23andMe is a good resource. It's about 80 to 90% accurate, but for $99, it gives a really good picture and it shows a lot of these SNPs. I have more information on my website. I have a free quick start guide. And so you can download that, and it talks a little bit more about testing. But I think there's a lot of testing that you can do. Like Dr. Amy Yasko's, I think is $500. There's ones that are thousands of dollars. But I really send my clients because they're usually short on cash, and they've done a lot of testing already for the $99 one, and then you can get that analyzed for like either free to $20, and then it just gives you a printout of all these SNPs.
All right, thank you. I know there's been some complications around 23andMe and people being able to get it analyzed can seem to be a sticking point, but we can talk a little bit more about that too, yeah.
Yeah, I'll just say the FDA stopped the 23andMe from giving interpretations, and I agree with that because as we just learned, we're all so individual, and it's not just our genetics alone. It's so many things that we have to take a look at, and so some of them, they would just be like, oh, well, you are 50% chance of getting cardiovascular disease, and they were only looking at a couple of the SNPs, and they're only looking at certain research. You have to really look at the person as a whole, but the testing itself and the SNPs were okay. It was the interpretation, and now they've actually progressed a lot further, and they're now working with some pharmaceutical companies, and they're opening that up a little bit more.
All right. Well, Genetic Solutions Now is a company doing genetic testing with Dr. Roberta Klein and Dr. Joe. They're going to do a couple webinars for us in November, not so much focus on MTHFR, but broader focus on genetic testing. So take a look at that. So when you say to look for the big four, the cardiovascular, fertility, cancer, mood, and then thyroid, autoimmune, are you suggesting that if somebody has all of these things, or if there's a family history of any of these things, that it might be something for them to consider testing?
I think it depends on the severity. I would look at the bigger picture. If there's a lot of, most all of my people, like I'll see like a huge thing of cardiovascular disease, and then they're having thyroid problems. They're also having like chronic fatigue. There's usually more than one big thing that's happening. And I'll just briefly mention usually pregnancy is one of the first times that I see clinically that people are having a problem. And miscarriage is usually one of the first kind of kickoffs of MTHFR, and it's because of that the increased B12 and folate need. And unfortunately, a lot of the prenatal vitamins have the folic acid, that synthetic folic acid that Mira was talking about, rather than the bioavailable forms of folate. Yes. And so that's usually the first one that I see people are causing problems with. But yeah, I think it's individual. I would just kind of look at it as a whole and, hey, yeah, this runs in my family and I'm feeling this, and I might only have a couple of these, but there's a lot of connections with family members, then I would probably try to get the testing.
Okay, so it's to look at these major kind of disease factors, and then the big picture of the individual. Once you do an intake, is to get a sense of their overall health pattern currently and over time.
Absolutely.
To see some kind of a pattern emerge.
Yeah, and I think one thing is very complicated clients. Like you're trying everything and things aren't working. Then that's one way to look into genetics. Then that's a good way to this is a tool that I can use. And even in my practice, I only really see people with MTHFR and methylation defects, and they've already seen a lot of practitioners. So I'm just, I look at everything as a whole, and I'm only using this as a tool. It's not the be-all and end-all of everything. I'm seeing which one of these SNPs might be causing a problem. And how we can create it. So yeah, just really complicated cases when clients are just not getting better.
Okay, thank you. And then you talked about people having detoxification problems and caution around that, and I really appreciate that because people are so bold around detox, and you know, for years we've seen it in our profession. People come in, it's like, oh, somebody's sick, you should detox. It's kind of automatic. People come in and say, I don't feel well, should I detox? And it's like, no, we're not starting there. And you know, just like you said, detox in a box is a big seller in supplement companies, and you have a big display in the store, and people come in and they buy a lot of it. And they go, yeah, I have detox problems. I need to detox. And
yeah,
so,
absolutely.
how do you know if someone is having a detox problem? And you mentioned that some people might have reaction to perfumes. Are there other signs that you typically look for?
You know, I my intake is 9 pages long, and then I have a couple other pages that they fill out, and then I spend 2 hours prior to the appointment reviewing everything, and then 2 hours with them on my first appointment. And so I ask a lot of questions on my form itself, and then I get into more details. Like I ask, like, where they grew up. Was it on a farm? Was it on an organic farm? You know, was it in the urban area? Were they near, you know, like, so I try to get a big picture of their exposure. Did they have secondhand smoke? Did they smoke? You know, because that's all of those heavy metals are present in smoke. You know, where they lived. You know, so I look at their exposure, getting all of those questions on exposure, and then I see like how, you know, have they done one of these detox in the boxes? And did they have a, you know, adverse reaction? Are they sweating when exercising? Are they able to, you know, function properly? Are they, you know, do they have chronic fatigue? Do they have fibromyalgia? Do they, you know, do they have all of those things? Kind of show that there's probably a detoxification. I hope to, you know, some people come in with the lab so I can see their glutathione or the antioxidant status. I ask them questions like if the, you know, the smells and the off-gassing, and does that make you nauseous? Does that give you headaches? And so I really try to get like the full picture of their exposure first, problems that may have come or diseases that may have come that have a connection with lowered detoxification, and then any history that they might have with doing some detoxification, etc. And then kind of put it as a bigger picture in patterns.
So I think it's really important you bring up back-to-back questions here following detoxification with testing. And back to that, if just because somebody has impaired detoxification or sluggish detoxification doesn't mean that they've got genetic SNPs necessarily.
No.
And so we can't assume that, but addressing and helping them support their natural ability to detoxify, you know, while taking all of these things into consideration, somebody should be able to improve pretty readily on a good nutritional protocol. And it's when we don't see them improving and continuing to struggle that we might consider branching out a little further and doing genetic testing. Is that your approach?
Absolutely, absolutely. And, you know, food, especially if they're eating a standard American diet, you know, I mean, we kind of know how destructive that can be. So just taking out, you know, that first step of taking out these additional toxins and then cleaning the air and, you know, making sure that they don't have a lot of off-gassing, all their products and all of those all the things that they're using on a daily basis or exposed to are being cleaned up. That just makes such a huge difference in a lot of people. And yeah, and it could be, you know, just nutrient deficiencies. It could just be the onslaught of their daily activities that they get more toxins in. And it could be SNPs, and it could be SNPs in the methylation cycle that decrease the glutathione and create inflammation, or it could be even, there's a lot of SNPs in the detoxification pathways as well. So there's a lot of reasons why detoxification can be impaired. So true.
All right. So coming here to nutritional deficiencies and diet and lifestyle, these kind of go together. We're talking about deficiency here of nutrients and a lot of factors that play into that. Really important information. But once you find that somebody is potentially deficient because of a variety of factors that are in place for depletion. Do you have any recommendations for specific nutritional deficiency testing?
You know, I like SpectraCell, although, you know, I'm not sure if it's 100% all the time. But I do like SpectraCell just because they test a little differently. What I find in like serum blood testing, it's showing more what's in the blood rather than what's happening cellularly,
Yeah.
especially with the B12 and the folate. So for example, like instead of the B12 serum, I like to go with methylmalonic acid and looking at that. And organic acid tests. If you, I'm not sure if your practitioners can order those, but those can be interesting as well, showing some of the metabolites, and then it can show that there might be deficiencies in some areas as well. So blood testing is not as great, you know, and that's why I kind of like SpectraCell. They show that what happens with the blood and with the nutrient itself and how that interacts with each other.
That's right. And SpectraCell and Genova Labs also has a panel called NutriEval.
Yeah.
And
Absolutely.
the thing about it that has benefit is that it shows enzyme functions as well.
Oh, nice. Yeah, and I can't order Metametrics, so SpectraCell is an easier one for me, but I love that. Yeah.
Yeah. We go with what we can order sometimes. Well, you mentioned diet and lifestyle. Was there specifics for diet? Any foods that you suggest that people eat more of or less of in general? Or does that really relate to the specifics individual?
You know, I think it's always bio-individual, but everyone can always eat greens. I know I forget who does it, but they're like, no, greens is bad, and I'm like, there's too much folate for COMT people. I don't think the folate from food can hurt us as much as the supplementation. And I think, the folate in like greens, in green leafies especially, green leafy veggies, is different forms of folate. So it's not all that methylfolate anyways. So I think as many greens as we can eat, and I think that's the number one thing that I try to do with my clients if they aren't already doing that, and then getting away from any of the processed foods. And I really just try to push people towards a whole foods diet, especially since I deal with a lot of very sick clients.
Right.
And to get all of that other stuff out. And, you know, gut health is so important as well, because if, you know, you can have the most perfect diet, but if you're not absorbing or digesting those nutrients, then that's going to be a major problem.
It sure is. It sure is. All right. Then there's going to be a number of questions coming up on testing, but I just want to come back to the individual SNPs because you talked about the gene cards. And there was a question about that. It's like, what are the gene cards and where do you get them? How do you do it?
Oh, Gene Cards is just a website, and it is really helpful because once you get those tests and you're like, whoa, I'm overwhelmed, I don't know what to do, you can actually put either the RSID number or the name of it. Like you could put MTHFR 677, which is there's several different MTHFRs, but the more prominent and the more studied is C677T and 1298C. So you could just put that name into the gene cards, and then it will list a whole bunch of information plus links to studies from PubMed.
Okay.
So it's just a way to kind of do more research once you have this information.
Got it. Do you have recommendations for amount of methylated folate, methylcobalamin, or anything else for somebody that's got MTHFR mutation and looking for preferred type and brands?
Yeah, so that's where it becomes really tricky because some people, and I look at the bigger picture with all the SNPs and which ones I believe are causing problems because methyl B12 can be very harsh with some people, but it is the most bioactive form of it. hydroxy B12. Usually most people can absorb that and take that. But your body does have to do more work to process it, and you do need the methyl nutrients to process that in your body. And then adenosine cobalamin is a little bit further down the pathway towards more in the mitochondria and energy production. So the hydroxy is what I will suggest to a lot of my clients who have mood and anxiety disorders. And that's kind of my big tip-off. I also look at SNPs. I look a lot of big things, but my big tip-off, if they have mood and psychological disorders, then then methyl B12 might not be the best for them. So sorry to simplify that and not give the full answer, but it gets very complicated.
No, it's an important. You had to answer it that way. It's very challenging to give just generic recommendations for when it's so complicated and we're dealing with just multiple issues often.
Yeah.
And so it's just out of respect and good nutrition practice. So thank you. What about this? Just in terms of statistics, this person said they've heard that at least 50% of people have one MTHFR mutation. It can be as high as 85%, and 10% can have both abnormal SNPs. Have you heard statistics like this?
Yeah, and it depends on the study. It depends on the population because different populations are higher and some populations are lower. So I think the percentages are just so hard to really. But a general view, up to 50% can be heterozygous, meaning they have one SNP that is SNPed, and then the other one is wild type or normal. And about up to 25%, I've seen everywhere from 15 to 25% can have two. And there's a lot of different variations of the 1298 and 677, but a big part of the population does have at least one of those, especially when you're combining them all together. And when you look at populations like Italians and Mexicans and Japanese, those are pretty high, high that have that SNP.
Okay. So when people have gut health issues also, do you have recommendations for determining what the status of their gut health is? Other tests or ways that you go about that?
I know there's a lot of tests for like leaky gut, and Cyrex Labs has some interesting testing, but I usually have my clients that come in with a lot of testing, and I don't do a lot of testing in my practice because by the time they've come to me, they've seen a lot of practitioners, and they're pretty deplete with monetary funds. Yeah. So I don't do a lot of testing for gut function. I look at inflammation. There's a lot of gut SNPs that I look at, and then I kind of rely a lot on just my intake form and my intake, just really asking them questions.
Sure.
And looking at it.
Depending on the situation, it may be relevant to do some permeability or leaky gut or food reactivity, sensitivity, intolerances, some comprehensive digestive stool analysis would be, the gold standard to really get a sense of dysbiotic factors and, pH
Absolutely.
and inflammation, so many things that can play.
Yeah, the Genova 2200, the comprehensive, is like the one that I would recommend more than any, especially if their gut, they've been to foreign countries and all of that, and symptoms are showing. Definitely stool testing, but there's a lot of different testing that you can do, and there's a lot of money that you can spend.
That's right.
And I think that we can do so much just kind of understanding what's going on, talking to them.
We can really get lost in testing world unnecessarily.
Right. And it could be
really helpful and really very appropriate. Have you see people that have used ACV, and I'm sorry, I'm not familiar with what that is. So see people that have tried this, but they're totally fatigued, and do you think it's healthy? I find it's a destructive thing as far as people are concerned with enzyme destruction. and how do you address this topic with the people you work with?
So I'm not sure, is it ACV like apple cider vinegar or?
Yes. Yes.
Oh, I don't think it's damaging for, I think it's great, for hydrochloric acid production, because some people just can't take HCl tablets or betaine. I don't know about enzyme destruction of apple cider vinegar. I've never heard that before. As far as the enzymes in the cell that we're talking about that speed up our chemical reactions rather than the enzymes for digestion, I don't think it affects that at all. I think apple cider vinegar could be harsh with some people, that have enough acid. I'm not sure. I think the question needs to be longer, or I think there's a lot of variabilities, if that makes sense.
Yes. Saying that all her children and herself have tested for MTHFR mutation. We're all positive with one of the mutations. One of my sons had both. My husband's not been tested. Can it be assumed that he likely has the mutation to contribute that to the picture?
Yeah, if the kids are homozygous, meaning they have two, then the father definitely has one.
Okay.
It's hard to go back and forward. We can say, oh, someone has one, they have this. It's harder to go back and forward. We can know for sure the people who are tested. If you're homozygous, then your parents each have one, and your kids will have one at least. That's what we know for sure.
Passed on automatically?
Yeah, so the genes are always passed on. These genes are passed on from our parents. Epigenetic triggers can be passed on as well from like famine or times of war, but that is more not, our DNA is passed on always.
Okay. And then a few years ago, a genetic test showed that they had hereditary hemochromatosis, homozygous. Okay. And then recently had a BPGEX genetics test that revealed they had a double mutation on MTHFR gene. Could there be a connection with these two epigenetically?
I haven't seen anything with like a connection because they're two different genes, but yeah, obviously they kind of have some of the same processes. So genetically there's no connection because they're two separate genes, but there can be some cross-section of problems that could occur.
Okay. In general, do you recommend probiotic supplementation? Is there any counterindication if someone would have a defect?
No, there's some that are better for histamine if people are having histamine problems, and methylation can cause higher histamine. There's some SNPs that can that degrade histamine, the DAO, that if they have those SNPs, they have problems with that. And then some people, obviously with other gut infections like SIBO, some of the probiotics. But I think probiotics in general is pretty good for most people.
Yeah. And then back to the greens that you were recommending, do you generally distinguish between which ones are high or low in oxalate content and to avoid inadvertently causing another issue? Some are extremely high, such as spinach, beet greens, curly kale, and Swiss chard, while others are much lower in oxalates, such as dinosaur, lacinato kale, bok choy, lettuce, and cabbage.
Yeah, it depends on if people have a history of I can't think of the name of that oxalates cause. The little balls that come out that are painful. I can't think of the name. The oxalate
kidney
balls.
stones.
Kidney stones. Thank you.
It raises uric acid.
When my mind is not there, I can't get the word.
Yeah, it raises uric acid too. So
I need some acetylcholine right now. So if some people are having problems, if they have a history of that, definitely, then I would probably do more lower oxalates. Other than that, I just tell people to try to kind of rotate their greens so they're not eating the same ones all the time.
And there'll be a difference here of cooked versus raw, do you think?
Yeah, cooking obviously destroys some of the nutrients. as far as folate goes, so I try to do them raw. If they have gut health problems, then I have them like try to pre-digest them, like in a smoothie or like in a pesto if they're gonna, and really just jam it out in the Vitamix or in the food processor.
But not steam it or cook them to reduce oxidation.
Steaming is okay. It's whatever you can do. If you like, what I tell people is like if you're doing if you're cooking them with other foods or if you're doing a soup, just throw them out the last couple seconds, so the least heat that they can really a high heat that they absorb, then the better the nutrient status.
So kale's been kind of the miracle vegetable of the year, I think, in terms of green drink promotion. And recently there's a couple studies and concerns that came out saying that, it's really a mistake that everybody's on kale because it, these leafy greens, particularly kale, really concentrate heavy metals. It's like, well, yes, they do.
Yeah. But so does everything. Why stop this great nutrient, that you can get in? And I read some debunking articles on that as well, and people wrote about it as well. I think sometimes, and I don't even know why, but it's just like if there's a food trend, then they're like, oh, we have to take this food trend down. Kale is such an amazing nutrient. Yes, some people can't handle it, but I think all in moderation is great.
Right.
Rotating your greens is always a good idea.
Right,
We're inundated with metals everywhere. It's in our soil, it's in our air, it's in our water.
All right, I think this brings us to a good closing point. We've got a lot to cover in 2 weeks when we come back with discussing this in relationship to more mood. But we'll talk about that here in a minute. Let me just go to some closing comments, if that's okay, and thank you for everything you've done here so far.
Oh, thank you so much. I appreciate it.
All right, we'll come back and talk about your upcoming one here in just a minute. But I want to let everybody know that there was a lot of information that was shared here, and I'm glad that we recorded it and let you know that it'll be available on Hawthorn's website under our archived webinars in just a few days. And you'll be able to review it, and there's a slew of other topics up there from previous webinars for all your viewing pleasure. And I just really hope that we've inspired you to learn more about health and nutrition. If you want to learn more, we offer it here at Hawthorn, and if you'd like to learn more about the programs and courses that Hawthorn offers, please visit our website. A lot of detail and information there, and Kathy McDermott is our Director of Admissions, and happy to talk with you. And as I said before we started, there's a survey to fill out when the webinar ends. It always helps us to have your feedback and your comments. We consider it carefully, so we sure appreciate you taking the time. And just a reminder that our webinars, Hawthorn's Holistic Health and Nutrition public webinars, are the first and third Tuesday of the month. All you got to do is reserve your spot, and if there's others that you can think of that would benefit from our educational webinars, you're more than welcome to share the information. When people join our mailing list, we'll let you know about our upcoming presentations. And here's a chance to let you know about Coleen's next one on September 1st for part 2 on the presentation of Are Your Genes Depressing You? MTHFR and Your Mood. And we're not going to talk about blue jeans here. And we'll start at 3:50 for our announcements, and we'll do a book review conversation, and we'll have Debbie Vale back with us for that. So I hope you join us for that informative segment, and it'll be a good presentation to tune into for sure, so come on back for Coleen. All right, this concludes today's presentation. Thanks again, Coleen.
You're welcome. Thank you so much for having me. I appreciate it.
You bet. And thanks to everybody for sharing the educational experience with us. Wish you all the best of health. I really look forward to coming together again in 2 weeks for Hawthorn University's next webinar. Until then, take good care.